Good afternoon, and it's a great pleasure to be here at the Alliance of Regenerative Medicine. So thank you to ARM for the opportunity to introduce our company and also for your efforts in fostering collaboration in the field. So this is the first presentation of Orchard Therapeutics at ARM. And so the main focus of my presentation is actually to introduce the company and to tell you who we are, what our pipeline is, and what we are about. I have also some information on some of the clinical data that we have generated, but the main objective is very much that you get a sense of what Orchard Therapeutic actually is. So this is a brief overview of our company. So Orchard is a therapeutic company with preclinical and clinical programs. The company was launched earlier this year, and our focus is on ex vivo lentiviral autologous gene therapy. Our therapeutic focus is on primary immune deficiencies and inherited metabolic disorders. So this is what defines our scope of activity. And one of the things that makes Orchard unique is that from the inception of the company, it was built on a number of collaborations with leading experts in gene and cell therapy research. So some of these experts are based in the UK, at University College London, Great Ormond Street Hospital, at the University of Manchester and Manchester Hospitals. Others are in France, Geneton. And finally, some of our collaborators are here in the United States and include Boston Children's Hospital and University of California, Los Angeles. And we see these collaborations as a great asset for the company. One of the other things that define Orchard is that, as I mentioned earlier, we're a clinical stage company. And the pictures that you can see here on this slide are pictures of patients who have been treated with autologous lentiviral gene therapy. So as a company, we're very fortunate to work on medicines that already make a great impact on the lives of patients. And this clearly gives us, an enormous motivation and impetus to go forward as we try to solve the many challenges associated with implementing such clinical programs for this technology and to try to make these medicines available to patients globally. So just a few words about why we are focused on autologous ex vivo gene therapy. So most of you are very familiar with the history of gene therapy. So the point of this slide is not sort of to rehash the various stages of research of gene therapy. I mean, this is clearly beyond the scope of this presentation. But you will agree that, in the past 10 to 15 years, a number of sort of very significant, POCs have been delivered, in a number of diseases. And our contention at Orchard is that ex vivo gene therapy has played a very significant role and generated a significant body of exciting data in the clinic, including in inherited metabolic disorders such as metachromatic leukodystrophy or adrenoleukodystrophy, including hemoglobinopathies such as sickle cell disease or beta thalassemia, and including primary immune deficiencies. So this is, we believe that this area will continue to be very important going forward. And finally, I think it's important to acknowledge the approval by the European Medicines Agency of Strimvelis, which is a gamma retroviral autologous gene therapy for ADA-SCID. I was fortunate to be part of this program when I was working at GSK, and I can really attest that this has required a very significant effort of GSK to get this approval over the line with the EMA. We see this as a very positive signal at Orchard, and it encourages us to go forward with our pipeline of lentiviral gene therapy products. So this is our pipeline. So as mentioned before, it is entirely defined by the technology of autologous lentiviral gene therapy. And we have two franchises at this point in time. So we have a franchise of programs in primary immune deficiencies and another franchise of programs in inherited metabolic disorders. So in PIDs, our lead program is with lentiviral gene therapy in ADA-SCID, which is often referred to as the bubble boy disease, and it stands for adenosine deaminase severe combined immunodeficiency. It takes a while to get used to the acronym, but you can refer to it as ADA-SCID. And in a moment, I will present a summary of the clinical data that has been generated to date in this disease. And then behind ADA-SCID, we have more programs with the same technology in primary immune deficiencies, which to date I cannot disclose. As far as metabolic disorders are concerned, our lead program is in an advanced preclinical stage. It is in a disease referred to as Sanfilippo type A, or MPS IIIA. Some other companies here at this conference are also working on this disease, so you've probably heard that this is a very serious life-threatening condition in which more than half of children and adolescents will die before the age of 20. And this disease is sometimes referred to as a childhood dementia, so a very significant and very worthwhile condition to address. And finally, we have a number of preclinical programs in similar diseases to complement our inherited metabolic franchise. So all of these diseases have in common that they are rare diseases. So they have incidences of between 10 and 100 patients in Europe and the United States, respectively, and prevalences of hundreds of thousands of patients. Now, our investment thesis as Orchard is that you can build a sustainable and prosperous franchise by aggregating several of similar diseases in areas of high unmet need around a common clinical development, supply chain, and commercialization platform. So this is what we are about. This is our mission. So the team that has been formed to advance these programs includes experts that have experience with gene and cell therapy product and with rare disease indications. And the colleagues here come from the industry, so Andrea Spezzi and myself come from GSK. Stuart Craig has been with a number of companies in the field, most recently with Sangamo. Our Chief Scientific Officer, Bobby Gaspar, is Professor of Pediatrics and Immunology at University College London. And Anne Duprat, our Chief Regulatory Officer, joins us from a contract research organization and has significant expertise in regulatory affairs in the field of gene and cell therapy. And she was actually involved in the development of EU regulations for advanced therapy medicinal products. So this is the team that has taken on the challenge to advance the programs we have in our pipeline. Our scientific advisory board contributes, I think, world-leading expertise in the disease areas in which we work in. Don Cohn and Bobby Gaspar have pioneered gene therapy in primary immune deficiencies, and the first patient, actually the first ADA-SCID patient to receive autologous gene therapy at UCLA was dosed in 1993. It's been quite a while. At UCL, the first patient was dosed in 2003. More recently, Professor Gaspar and Professor Cohn have actually transitioned to a lentiviral gene therapy approach, and the ongoing study, which I will present in a moment, has started respectively in 2012 in London and 2013 at UCLA. So this just illustrates the continuum of research and the successive improvements that those experts are making to the technology. I should also acknowledge Adrian Thrasher and Wasim Kazim, who have been involved in many clinical trials in primary immune deficiencies, and Fulvio Maviglio from Geneton was actually involved in the very first gene therapy experiment in ADA-SCID that involved the transfer of a gene into bone marrow cells in 1992. As far as our metabolic franchise is concerned, our collaborators in Manchester, Brian Bigger, Simon Jones, and Rob Wynn, have world-leading expertise in the treatment of metabolic disorders and gene therapy, and their approach with autologous lentiviral gene therapy builds on the seminal work of Alessandra Biffi in metabolic diseases, and some of you may be familiar with the remarkable results that were delivered with Alessandra Biffi's program in metachromatic leukodystrophy. So this is an approach that builds on the same paradigm, if you will, in a related disease. And Alessandra Biffi has recently joined Dave Williams in Boston, who is a world-leading expert in hematology and oncology disorders. So this scientific advisory board is an asset for the company. It is poised to be a significant engine of the growth of our pipeline, and also it is naturally going to be to provide partners for the clinical development of our medicines as they transition from the clinical phase into the clinical phase. So what data has actually come out of these collaborations? So given the time that we have for this presentation, I will present top-line data from a lead program in adenosine deaminase severe combined immunodeficiency, or ADA-SCID. So this is a rare primary immune deficiency. So it's a monogenic disease caused by one of several mutations in the adenosine deaminase gene. And these mutations result in a deficiency in the enzyme adenosine deaminase, and that in turn leads to profound lymphopenia, T cells, B cells, and NK cells. And as a result of this, children have a profound immune deficiency and are susceptible to life-threatening infection. And without treatment, a child with ADA-SCID will die within the first year of life. So this is a rare disease with an incidence of about 20 to 40 patients in Europe and the United States. And we're actually finding out over time what the real incidence is as newborn screening is being rolled out in now a vast majority of states in the United States. I think it's now more than 40 states, and also in a number of other countries across the world. But it's taking time to roll out newborn screening. So now we have an ongoing trial in this disease, and at this point in time, a total of 39 patients have been treated. The most recent statistical analysis that we have conducted dates March 31, 2016, so about 4 or 5 months ago. And all patients have survived, so we have 100% survival. Event-free survival is when a patient who has received gene therapy is free of enzyme replacement therapy and does not require a subsequent transplant. So we have 100% event-free survival in our US cohort. That's 18 patients out of 18. And in the UK cohort, we have an event-free survival of 93%. That is 14 out of 15 patients. So the one patient who did not meet this criteria was a patient who responded very well to enzyme replacement therapy, and therefore the gene-modified cells did not engraft. But for all of the patients, the gene-modified cells did engraft and did reconstitute the immune system of the patients so that they were free of supplementation with enzyme replacement therapy. And what gives us great encouragement is that we've observed a stable persistence of gene-modified cells in these patients. And therefore, we anticipate that the benefits of treatment will last over time, and this opens the prospect for a lifelong cure of patients. So I wish I could show you some pictures of patients that have received this treatment, because you can really see the striking benefit of gene therapy, but for confidentiality reasons I can't do it. But I would like to mention a very recent example of a Lebanese boy that was treated at UCLA. The boy was hospitalized at the age of 6 months. Was in a critical condition. The patient was weighing 4 kilograms, which puts him well below the 5th percentile for its age group, had a very serious respiratory infection, and required continuous airway positive pressure. So that child was first initially put on enzyme replacement therapy to stabilize the patient, and that has allowed also the child to gain some weight. So that boy was dosed with gene therapy mid-July. At the age of 10 months, he received 15 million cells per kilogram. And the patient actually was discharged pretty much exactly 1 month after receiving gene therapy. And at that point in time, also, Professor Cohen was able to stop treatment with enzyme replacement therapy. And for the first time in his life, the boy was able to eat solid food. So this is a great story. And every week, every month, we receive sort of summaries, and we see pictures of these patients, and it's very, very motivating and encouraging for us. Now, I should also mention, and my presentation would be very incomplete if I did not acknowledge the challenge of building a global integrated supply chain that delivers these personalized medicines to patients wherever they are in the world. This is a very, very significant challenge. It's actually a twin challenge of first implementing a supply chain that will meet commercial GMP requirements. And we have a titanic work to do to transition from the current sources of supply to commercial GMP requirements. That is a major challenge. The other challenge is to deliver cells to patients wherever they are, if you like. Our aim is that a patient can receive treatment in a transplant center near where they reside, and that involves cryopreservation. So these are considerable tasks. Really, they're not trivial. And we cannot do this alone. So we stand on the shoulders of expertise, of CMOs that have expertise in gene and cell therapy. And really together we want to master this challenge. This is going to be a key area of focus for the company going forward under the leadership of my colleague Stuart Craig. And it's very obvious that we're not building this platform just for one medicine for our ADS kit program. Our aim is to build a platform that can serve all of the programs in a pipeline and therefore to realize economies of scale. So I hope I've given you a sense of our company and our focus, and I hope you will agree that we have sort of great ingredients for success. But our challenge as a company now is very much in execution, so in advancing the programs to clinical development, in having a judicious regulatory strategy for programs, in particular for preclinical programs, in building a robust supply chain globally, and of course to build an organization that will commercialize these medicines and gain access for this treatment. So therefore we are very, very keen to go rapidly as an organization, and so we are hiring across the board. So if you are inspired by what we do and you would like to work in an environmental and a challenging and entrepreneurial environment, please do let us know. So thank you very much for your attention.